Hello everyone, we are going to discuss immunity. Immunity means protection from disease. So it is the recognition of non-self and its subsequent destruction. Immunity is the responsibility of certain cells and molecules that constitute immune system. So immune system has the responsibility to develop immunity. And their organized and coordinated response to an invading foreign substance is known as immune response. So types of immune system. There are two types of immune system, innate and adaptive. Innate immune system is also known as natural or native immune system, and adaptive is also known as acquired or specific type of immune system. So innate system, it has cellular and humoral immunity. Cellular immune system or cellular immunity, it has components macrophages, neutrophils and NK cells, while the humoral system has complements. The cellular component of adaptive immunity is lymphocytes, T lymphocytes, while the humoral include B lymphocytes and the antibodies produced by B lymphocytes. So coming to the innate immunity, it is the first line of defense and it has following components. Number 1, epithelial barriers. Epithelial barriers, they act as physical barrier and block the entry of microbial agents. Epithelial antimicrobial peptides are defensins, cryptocidins, hystatins, and granulysin. Next component is phagocytic cells. Phagocytic cells are neutrophils, macrophages, and natural killer cells. Then the plasma proteins. Plasma proteins like complements C3A, C3B, C5A, and membrane attack complex comprising of C5b, 6, 7, 8, and C9. Then C-reactive proteins, mannose binding lectin, ceruloplasmin, haptoglobin, and alpha-1 antitrypsin. Adaptive immunity. The immune response that develops in response to exposure to infectious agents, and it increases in magnitude with repeated exposure to the same agent. So adaptive immunity is basically developed in response to exposure to infectious agents, and its intensity or magnitude increases with repeated exposure. So we are going to differentiate innate immunity and adaptive immunity. Innate immunity, also known as natural or native immunity, while adaptive immunity is specific type of immunity. Innate immunity is the first line of defense, while adaptive immunity appears after innate immunity. It develops in response to microbes and not to non-infectious substances, while the adaptive immunity, it develops in response to very large and diverse spectrum of infectious agents and molecules. Innate response. In innate immunity, the response remains the same. It is very important. The response remains the same even with repeated exposure, while in adaptive immunity, we have already discussed in previous slide that response increases in magnitude with repeated exposure by the same agent. In innate immunity, no memory is formed, while the adaptive immunity, it has memory. In innate immunity, unable to differentiate between related microbes and molecules, while the adaptive immunity, it has the ability to differentiate between closely related molecules and microbes. That's why it is specific immunity. And the component of innate immunities are epithelial barriers, phagocytic cells, and plasma proteins, while the component of adaptive immunity are lymphocytes and antibodies produced by lymphocytes. So it is important to differentiate between innate and adaptive immunity. So we are going to discuss now major histocompatibility complex. Histocompatibility antigen is group of antigen displaced on the surface of donor cells, which is recognized by recipient's immune system, leading to transplant rejection. So histocompatibility antigen is present on the surface of donor cells and it is group of antigens. Each individual inherits a set of unique alleles which cause transplant rejections. Hence, they refer to as major histocompatibility complex genes. MHC genes, they are also known as human leukocyte antigen because initially they are detected in leukocytes. So, human leukocyte antigen HLA. Major histocompatibility complex genes, they are clustered on small area of chromosome 6. So, they are present on chromosome number 6 on the small area in 400 kbp long stretch. So, that small area on chromosome 6 is major histocompatibility complex. Proteins encoded by MHC genes, they express their antigens in a codominant pattern. Within the MHC, major histocompatibility complex, there are 3 blocks of genes. So MHC on chromosome number 6, there are 3 blocks of genes in the MHC major histocompatibility complex. Class 1, class 2 and class 3. So first we will discuss MHC class 1. It encodes a glycoprotein which is expressed on surface of all nucleated cells. So the site is on the surface of all nucleated cells like B cells, T cells, platelets, and it is absent in RBCs. So the structure of MHC class 1, it consists of 2 polypeptide chains. Heavy chains alpha 1, alpha 2 and alpha 3. This is heavy chain that passes through the plasma membrane. Then the light chain, which is beta 2 macroglobulin. MHC1 has 3 closely linked loci, HLA-A, HLA-B and HLA-C. So, class 1, it has heavy chain and light chain. Heavy chain comprising of alpha 1, alpha 2, alpha 3, while beta 2M form the light chain, and it has 3 closely linked loci, HLA-A, B and HLA-C. Function of MHC class 1 is binding and display of viral proteins and binding and displays of cancerous proteins. So viral proteins and cancer proteins, they are bound and displayed by MHC class 1. So coming to mechanism of action of MHC class 1, viral genome, it is inserted into host DNA leading to production of viral proteins, then broken down by proteolytic complex to short peptides. These viral proteins are broken down then transported to endoplasmic reticulum, then binds to antigen binding site on MHC1, and further they are transported to cell surface and presented to CD8 plus cytotoxic T cells. MHC class 2. Gene encodes a glycoprotein which is expressed on antigen presenting cells, macrophages and B cells. So they are present on macrophages and B cells, and they present the foreign antigen to T helper cells, CD4 plus cells. So structure of class 2. Class 2 genes are arranged in 3 loci, DP, DQ and DR. They are also membrane bound. Alpha chain, extracellular part, alpha 1, alpha 2 domain. There is one transmembrane part and cytoplasmic part. Extracellular part, transmembranal part and cytoplasmic part. Alpha 1, alpha 2 domain, they are extracellular part. Beta chain, it also has extracellular, transmembrane part and cytoplasmic part. Beta 1 and beta 2 domain are extracellular part. Alpha 1 and beta 1 domains, they interact to generate an antigen binding site. Function of MHC class 2 is binding and display of foreign proteins to CD4 cells. So mechanism of action of class 2, MHC class 2. Microbes or soluble proteins, they are engulfed by macrophages and processed in endosomes or lysosomes. They are cleaved by proteolytic enzymes into shorter peptides, which are then transported to endoplasmic reticulum. Then they binds to APC, antigen presenting cells, on MHC2, and they are presented to CD4 plus helper T cells. So you can see the picture in which viral infected target cell, the viral proteins, they are engulfed or attached to MHC class 1, and this MHC class 1 will present it to CD8 T cells. And while MHC class 2, in this antigen presenting cell, antigens are bound to MHC2 by antigen presenting cells, and MHC2 will present it to CD4 T cells. Now MHC class 3 genes, they encode for components of complement 4, 2 and factor B. They do not act as histocompatibility antigen. Now the diseases associated with HLA types are inflammatory diseases, autoimmune diseases and insulin dependent diabetes. So inflammatory diseases like ankylosing spondylitis, acute anterior uveitis and post-gonococcal arthritis, they are associated with HLA-B27. So HLA-B27 is associated with these inflammatory diseases. So autoimmune diseases, they are also associated with HLA types. Systemic lupus erythematosus, it is associated with HLA-DR3. Rheumatoid arthritis associated with HLA-DR4. Myasthenia gravis, it is associated with HLA-DR2, and the insulin dependent diabetes, it is associated with HLA-DR3, HLA-DR4. Thank you.